antigen-presenting cells? Where are macrophages and mast cells found? What paracrine do basophils and mast cells secrete? Go. That's 5 questions for the price of 1 right there. I'll give you a break. Those who have entered their answers, for their sake, what white blood cells can phagocytize pathogens? Yes, they're called phagocytes, and they include neutrophils, eosinophils, and there are monocytes in the blood and macrophages in the tissue. Which ones are cytotoxic? They can kill infected cells or cancer cells. That would be the cytotoxic T cells, which are lymphocytes. And also, eosinophils are cytotoxic. They can kill parasite cells. Now, this is a new term. Which ones are antigen-presenting cells? Antigen-presenting cells phagocytize something or somehow get the protein of a pathogen inside their cell. Then they expose it on their cell surface, holding it out like a hand holding out a hot dog. On their surface, presenting the antigen for other cells. The 3 types are the dendritic cells, the lymphocytes and plasma cells, particularly the B lymphocytes, and the monocytes, macrophages, which are grouped together. Monocytes in the bloodstream, macrophages in the tissues. They're actually not the same type of cell in 2 different places. They're slightly different cells in 2 different places, but they behave very much alike, and they're called the mononuclear phagocyte system. These macrophages are all around in our tissues waiting to chew up pathogens that get under our skin, but not into the bloodstream per se. That answers the next question. What paracrines do basophils and mast cells secrete? Histamine, which mediates the inflammation and allergic response. The mast cells, we've talked about being under the skin and producing itch. Basophils are similar types of cells in the bloodstream. The most common white blood cells, though, in the bloodstream are these phagocytes. The neutrophils, also known as polymorphonucleocytes, multiple shaped nuclei. And that gives meaning to this mononuclear word. The mononuclear phagocytes and the mononuclear lymphocytes have simple nuclei. These others have complex nuclei and vesicles. neutrophils have both eosin and hematoxylin binding vesicles, whereas eosinophils their vesicles bind eosin and basophils bind hematoxylin. So these PMNs, polymorphonucleosides, sometimes are called poly, sometimes they're called segs because they have a segmented nucleus that's pretty easy to see. When they're immature, they're called bands or stabs by the lab tech that is doing the diff, the count of how many different types of white cells there are. So let me back up just a little bit and mention that there's different types of pathogens. Pathogens are anything that cause illness, pathos, generate those, including bacteria, viruses, fungi, protozoa, parasites. Pollens cause allergies, they're pathogenic in that sense. Chemicals can injure us, they're pathogenic in that sense. Foreign bodies can get stuck and cause an immune reaction and scarring. They have in common that they contain antigens, which are molecular surfaces that can be recognized by the immune system. And these surfaces are mostly small peptides with a certain amino acid sequence that has therefore a certain surface shape that antibodies can bind to. I've been talking a lot about the virus working a lot on the virus influenza in my lab. I won't take time this morning. We don't have time to talk much about it, but we have kibitzed a little about its relationship to coronavirus already. Let me just say this. It's intriguing to me that everybody's been prepared for an influenza pandemic, and we got hit by a much more subtle and devastating pandemic, because influenza is easy to identify and it identifies itself quickly. Within a day or two of getting infected, you already are knocked off your feet. But coronavirus is the cold that kills. It comes on subtly, takes a long time. You might be a carrier and not know it. And all of a sudden you get SARS, severe acute respiratory syndrome, from it. This coronavirus, they call it SARS-CoV-2, that causes COVID-19, is a cold that kills. Usually these pathogens get swept up through our lymphatic system, passing through lymph nodes, where they can be presented by dendritic cells in the lymph nodes that bind up the pathogens and then present their antigens to white blood cells. The same happens in the spleen, especially the white pulp of the spleen. And these dendritic cells you don't hear about very much because we talk about the white cells in the bloodstream. These are ones that are housed in the spleen, in the lymph nodes that are named for having lots of spines on them, and they are antigen presenting cells. They're like the housekeepers in our lymphatic system. Next question, question 2. Innate immunity may be saving many people from COVID-19 infections, especially through interferons in the cytoplasm of infected cells, natural killer cell induced apoptosis of infected cells. I left off the on killer accidentally. Cytokine release to bring more phagocytes and natural killer cells to the infected region, and virus phagocytosis by macrophages. That's innate immunity. Those are many of the innate responses. What is the hallmark of acquired immunity, which a patient will have after an infection, or they can get by vaccination? Ryan Sorensen, can you tell us what you think the hallmark of acquired immunity is? I trust that Ryan can't speak. Maybe his mic isn't connected. Amber Sessions, can you?
Yes. So what I was thinking was just like the T cells and the B cells. So with the acquired immunity, it kind of is like you, I guess in a way, like build up an immunity to it. So like your T cells and your B cells, they would, in a way, like, I think, because the B cells has like memory cells or something like that, and so they can remember like the pathogen, so then they can like attack again the next time it comes. So I guess you almost like your body gets acquired to it, and then I guess.
Excellent job, Amber. And what is the molecule that they use to recognize the antigen? Do you remember that? B cells and T cells. Antibodies, right? So antibodies are a key molecule in the B cell and T cell that make them the hallmark of acquired immunity. Other aspects of innate immunity include our skin keeps out infections, our phagocytes, our macrophages are sort of general chew them up and eat them up, anything foreign types of cells. We have the C-reactive protein and complement cascade. We're going to talk about more in a minute. Opsonins, which are generally antibodies that activate the complement cascade. Histamine, released by the basophils and mast cells. Pyrogens, which can destroy cells by being spat from one cell into another. All of these are part of the inflammatory response. Plus we have chemotaxins like cytokines, and our mucosa, including peptides in our skin and mucosa that can kill. And also passive immunity can be transferred from mother to baby through mother's IgA and IgA via the womb, the uterus, and IgG via the uterus, and IgA via the milk. Question 3, some particles like viruses can be directly phagocytized by macrophages and others. But bacteria with polysaccharide capsules must be opsonized first. What component of an antibody bound to a bacterium can be recognized by a phagocyte to stimulate phagocytosis? Austin, can you tell us? No? Marin, can you tell us?
I just put the FC region.
Of what?
Of the antibody.
That's right. Opsonins are typically antibodies, and it's the FC region of an antibody that sticks out from the bacterium because the FAB region will bind to the bacterium, bind to antigens on the bacterial surface. And then the FC pokes out. And then the phagocyte can bind the FC region and like this, well, no, like this, that's what I want to point to. And that induces phagocytosis of the bacterium. It's the polysaccharide capsule that makes the bacterium somewhat immune to other kinds of phagocytosis, where the macrophage or other phagocyte recognizes antigens directly through proteins that stick off of its surface. So opsonization involves facilitating the activation of phagocytosis. Question 4, what do lysosomes use to digest phagocytized material? Luke, can you help us with that? If not, Jade, how about you? No, Jade?
Hello? Can you hear me?
Jade, are you there? Yes, I hear you.
Yeah. All right.
What do you say lysosomes use to digest phagocytized material?
I said hydrogen peroxide, just like, what's the other one? Oxygen and NO.
Right, NO, nitric oxide, is a free radical, I guess, that helps break down other molecules. Superoxide, O minus, is a free radical. And hydrogen peroxide can make free radicals easily. And these, including these and enzymes, break down things that have been ingested into phagosomes. And then the phagosome fuses with the lysosome. These lysosomes have been in the news lately with coronavirus because they become very acidic inside. They are, they have membranes as vesicles with proton pumps in them that pump hydrogen in. And they also have, for some, not for some reason, one of the enzymes that they have is cathepsin. Cathepsin is an enzyme in lysosomes that helps prepare coronavirus by cleaving its spike protein. And once the spike protein has been cleaved, it can then fuse itself with the lysosome wall and spit its RNA out into the cytoplasm to go to the nucleus and infect the cell. So cathepsin blockers would block coronavirus infection at this stage. Or if you can inhibit the acidification of the lysosome, cathepsin won't get activated because it's only active between pH 3 and 6, not pH 7.4. So what if you had some molecule that could get across the membrane easily and bring protons back out as fast as they were getting pumped in? Guess, can anybody tell me what molecule does that besides Morgan? Go ahead, anybody chime in. Go ahead, anybody.
Could you repeat the question?
What drug makes the lysosome neutral by carrying the protons back out as fast as they're being pumped in? I'll give you a clue. It's used for malaria. Oh, you guys, don't you even watch the news?
Was that the hydrochloroquine?
Yes, hydroxychloroquine or chloroquine, which are bases, hydrophobic bases that can easily diffuse across the membrane, pick up a proton, carry it back out. Okay, question number 5. C-reactive protein binds to the lipid membrane of dying cells, bacteria, and parasites to initiate the complement cascade, where many proteins, more than 25 proteins can participate in this. They gather together, form aggregates. What does the complement do to destroy the unwanted cells that the C-reactive protein has bound to? Answer is complement consists of many molecules that gather together to make a pore complex. It's called the membrane attack complex, and its pore is so big that basically water can get in easily. The nucleus essentially, or the nuclear material, can't get out. So osmotically, they just draw water in and it swells up the bacterium until it bursts. Lyses.
Would it be possible for bacterial cells to try and regulate that difference, if possible, or remove?
They don't seem to have an easy way to regulate it because they're full of proteins, macromolecules that are too big to get out through the pore, so water comes in through the pore.
Yeah, so they can actually try preventing that pore from occurring. They have mechanisms to stop those proteins from adhering, but once it adheres, the bacteria is dead. So,
Okay.
That's a good question, and maybe you can chime on this one too, Morgan. I don't quite know the answer. How is it that they attack pathogens and not our own cells?
yeah, so it's actually one of the C proteins, specifically C3 or C4, has a pathogen like binding domain. So it will spontaneously hydrolyze to the active form in the bloodstream. And if it doesn't find like a lipid polysaccharide coat, then it just gets degraded. But if it does, it attaches and then starts the whole process. So it can also look for like LPS or any of those antigens that the acquired immune system is going to look for too.
Thank you very much. And are you referring to a component of C-reactive protein when you say C protein?
Yeah,
or just some other complements?
Yeah, so there's about 20 of them, of those C-reactive proteins. It goes all the way from C1 to C15 or 16, I think. And they all have a specific portion. It's like a cascade, like the blood clotting cascade. C3 is the one that usually first grabs onto the bacteria, and then we'll clip C4 and C2, and C4 and C2 clip 5. And yeah, it's just a whole long thing people have to memorize.
Kind of similar to the clotting cascade. Question 6. What are the two chains that make up IgG? It's kind of a trick question. But so you'll see why in the second question. What are the two components of the antibody, IgG, and which chains are they made from? Garrett, would you mind giving us your answer to that? Garrett's not listening. Garrett doesn't want to answer.
Yeah. So there's two different parts to Ig, or the immunoglobulin G. It's got the Fc region, the Fab region, and the Fab region is made up of the light chain, and the Fc region is made up of the heavy chain.
And the Fab region also has heavy chain, right?
Yes.
That's right. So immunoglobulins are these globulins that we refer to that are in the bloodstream, some of the globular proteins, but they're very characteristically Y-shaped with a heavy chain that forms the backbone of the Y, and then a light chain attached to the heavy chain by disulfide bonds. And it's this kind of loose connection between the light and heavy chain at the end that is hypervariable, sometimes called the paratope, or is the antigen binding site. It's this part that has very specific surface complementarity to antigen surfaces. So it's just the shape of the antigen just fits into this hand like a hand in glove, into this glove like a hand in glove. There's two heavy chains. They're bound together by disulfide linkages, and they can be clipped at the hinge region, so you could have only Fc with two heavy chains attached by 4 disulfide bonds, and only Fab. But more importantly, they swing quite easily so that the Y can be broader or narrower. It's papain that cleaves them, a protease that can cleave them in. So the next question 7, yes.
Is it the light chain that varies greatly between like different antibody responses, or is it both the light and heavy chain that varies a lot?
Oh, that's a great question. My understanding is light chain. Who can tell better? Anybody know better than me? I think you're right. It's the light chain that has the hypervariable region on
Does it bind by like hydrogen bonding or I was wondering how that works.
Yes, hydrogen bonding and ionic bonding can come into play, but they're not as important as the van der Waals contacts of surface complementarity.
Okay, cool.
Next question, how many antibody molecules make up an IgA? How many make up an IgM? Where do you find IgA and IgM? And where do you find the monomeric antibodies? IgD, IgE, and IgA. Marin, what do you say? Can you help us with this?
Sure.
What's your answer?
So IgA is a dimer, so it has 2 antibodies. And these are found in external secretions. So saliva, tears, and then like mucus, breast milk, things like that. IgM, I believe, is a pentamer, so it has the 5 antibodies. And these ones are going to be found on the red blood cells, is what I found. And then you asked where IgD is. I'm pretty sure that's just going to be on the surface of beta lymphocytes. So that'll be like in the extracellular fluid. IgE is going to be in, again, ECF. And then, was it IgA again? Yeah, so like plasma and all those secretions.
Okay, good. Thank you. Going backwards over what you said, elaborating on IgE, it's in the extracellular fluid and binds through its Fc region to mast cells to stimulate degranulation in the allergic response. IgD, as you said, is in what you call beta cells, but I think you meant B cells. B lymphocytes express IgD on their surface in the secondary immune response. In the primary immune response, they secrete and express IgM, this pentameric antibody. So you might think in the primary response, you've got these super binders that could really bind things up, but they're not very mobile. They're all attached to each other. So in the secondary response, you have IgG. With IgD forming the receptor on the surface and IgG being secreted by plasma cells to just diffuse around in the blood, opsonize bacteria and other things. Great job, Maureen. Thank you. Calling you Maureen. I know you might not like it. Question 8. The secondary immune response is higher and faster than the primary immune response. What cells allow this more rapid response? Cameron, can you tell us your answer? No mic, Cameron? How about Craig? Craig Brailsford, do you have a mic? Can you tell us your answer?
Yes. So just technically memory T and B cells.
That's right. So we get the memory cells that produce the primary response, where antibody goes up quite a bit in the plasma, within a few weeks of antigen exposure, and then a secondary response where it starts going up right away upon antigen exposure. Because why? You already have memory cells sitting around producing antibodies, ready to produce antibodies Monoclonal antibodies are those that come from one particular cell. It can only express one type of antibody. If you have many different white blood cells that are all immune to the same pathogen, but at different epitopes or locations on the pathogen, different antigens of the pathogen, then we call that polyclonal antibodies. Multiple types of antibodies attacking the pathogen at different places. So it's these memory cells that continue to reproduce and are all ready to go upon secondary exposure that give us long-term immunity. That's why we get vaccinations. That's what we don't have to COVID-19. Name 6 roles of antibodies in fighting infections. Now, I wouldn't ask you that question on a test. Who's going to memorize 6 roles of antibodies? But Sarah, you did a great job of at least reading the slide. They activate B lymphocytes. They can act as opsonins. They can cause antigen clumping and inactivation of bacterial toxins. They can activate antibody-dependent cellular activity, like in the natural killer cells or the eosinophils, when they're exposed to antibodies as opsonins attached to the polysaccharide coat on a bacterium like E. coli. They can trigger mast cell degranulation, releasing histamine to attract more white cells to the neighborhood and relax blood vessels so that more blood comes to the neighborhood, relaxing the arterioles, smooth muscle relaxation. And they can also activate complement and the complement cascade. Question number 10. Major histocompatibility 1, MHC1 proteins, are used to present self-peptides on all cell surfaces, which means peptides that are found in our native proteome. But when they start presenting foreign antigens, the cell is attacked by cytotoxic T cells. So somehow the white cells know not to attack, that is the cytotoxic T white cells, know not to attack cells that are expressing self. Any that might be tempted to do that die, but when they're not expressing self, they recognize foreign and attack the cells. Why would normal cells present foreign antigens? And where are the cytotoxic T cells programmed? Luke, are you able to answer that question? Those two questions.
I thought that maybe, I don't know if this is right, but like if a cell was infected by a pathogen, then it would present non-natural cell markers. I don't know if that's
good. Perfectly done. They get chewed up in the lysosome. Proteins from the pathogen get chewed up in the lysosome, and then look, they get displayed on the surface. These surface molecules, MHC1 molecules, are actually like hot dog buns, and the peptides are like dogs that splay out from the two ends of the hot dog bun and are exposed by the antigen presenting cell. And then Luke, where do these T cells get programmed?
I think it's the thymus, right?
That's right, in the thymus, which is located where? In the mediastinum, above the heart, between the two lungs, in front of the trachea, below the thyroid gland, not to be confused with thyroid, right? Thymus is down in the chest, thyroid's up in the neck. And this thymus is part of the problem in people my age and older. It's getting worn out. It involutes. It becomes so small that in a cadaver you can't even find it oftentimes. So old people can't make their T cells. They can't program new T cells, so they don't have as good a T cell immunity, and they're very vulnerable to coronavirus attack in the case of SARS-CoV-2 that causes COVID-19. Question 11, specialized antigen presenting cells. Which ones again? Oh yeah, macrophages, B lymphocytes, and dendritic cells. These are the APCs, the antigen presenting cells. Macrophages, B lymphocytes, and dendritic cells all have major histocompatibility to proteins, a different, slightly different sequence of membrane protein that will present antigens. But this is on the white cells instead of every cell in the body. These antigen-presenting cells present foreign antigens derived from phagocytosis of pathogens, but their job is a bit different. In this case, their job is to present some other, stimulate some other T cell to secrete what? Instead of attracting cytotoxic T cells to kill themselves, they stimulate other T cells to secrete something. What is it? Cameron, can you tell us?
Sorry, did you say my name? It just cut out.
No, I said Cameron.
Okay.
But thank you. Thank you, Maureen. No, Cameron? The answer is these antigen presenting cells stimulate I don't know, I can't think of the answer. Stimulate helper T cells These antigen-presenting cells stimulate helper T cells to secrete cytokines that activate other immune cells. And that will come back up in just a moment. So this is what I was thinking of as I wrote that question. Question 12. In the classic virus infection scenario, what are the roles of natural killer cells and cytotoxic T cells? What are the roles of helper T cells and B lymphocytes? Answer is natural killer cells and cytotoxic T cells attack any of the host cells, meaning the whole person, all of the cells in the body of a possibly infected person, attack those cells and then inject perforins and granzymes in them to destroy them. So that's why they're called killer cells, and that's why they're called cytotoxic. They are toxic to the cell. They kill the cells that are presenting foreign antigens on their surface. How do those foreign antigens get there? By the cell getting infected and having the virus antigens chewed up by the lysosomes and then presented on major histocompatibility protein 1. What are the roles of helper T cells and B lymphocytes? Helper T cells activate B lymphocytes after being activated themselves by macrophages that are also presenting the antigen, but this time on their matrix compatibility complex type 2. These macrophages then wake up the helper Ts, the helper Ts go and wake up the B lymphocytes, turning them, converting them into plasma cells. Plasma cells are named plasma cells, I guess, because they're found in the plasma and also because they're secreting antibodies like crazy in the plasma. And these antibodies are free to diffuse around in the plasma. They're IgG in the secondary response, IgM in the primary response, and they can go back and bind up the viruses, preventing them from infecting other cells. And they also can opsonize the virus so that macrophages can ingest them more easily. So think of this as the virus acquired immunity mechanism. What about the bacterial acquired immunity? Question 13, go. In the classic response to a bacterial invasion, perhaps through the skin, what cells are activated by phagocytes, like macrophages, to stimulate B lymphocytes to produce antibodies? Answer is helper T cells present antigens to B lymphocytes. And stimulate them to secrete antibodies, which can opsonize bacteria, which can stimulate complement proteins to coat the bacteria and make them easier to digest by phagocytes that are embedded in the skin outside of the bloodstream or in the mucous membrane or in the extracellular fluid beneath the skin, being able to protect us from breaks in the skin that bacteria can get in through. Question number 14. In the allergic reaction, what cell is activated by antigen-presenting cells, APCs, and then stimulates B cells to convert to plasma cells so that they can make IgE? Then, what cell can IgE bind to by its Fc component and stimulate degranulation when it binds the original allergen? The answer is that helper T cells activate B lymphocytes, and that helps them differentiate, convert into plasma cells that can secrete antibodies like crazy, and they do secrete IgG, which produces inflammation through complement cascade, but they also secrete IgE. IgE is then picked up by the mast cells via its Fc
Well, I guess it's 9:50, so you guys can just head out. I'll just give you guys the digital dialogue points if you finish it. So, okay, have a good day.
I mean,